Hi everyone, today's video will cover three topics which are glycolysis, Krebs cycle, and electron transport. So, let us start with glycolysis. What is glycolysis? Glycolysis is the breakdown of one glucose molecule in a series of enzyme-catalyzed reactions into two molecules of pyruvate. The process occurs in the cytosol of the cell cytoplasm and does not require oxygen. A brief introduction of the main stages of glycolysis. There are 10 enzymes involved in breaking down sugar. The 10 stages of glycolysis are organized by the order in which specific enzymes act upon the system. The net product are 2 pyruvate molecules, 2 ATPs, and 2 NADH. In stage 1, the glucose molecule is phosphorylated with the help of enzyme hexokinase. Phosphorylation is the process of adding a phosphate group to a molecule derived from ATP. As a result, glucose 6-phosphate is produced. One molecule of ATP is consumed during this phase. The second stage of glycolysis is the rearrangement of glucose 6-phosphate into fructose 6-phosphate by the enzyme phosphoglucose isomerase. Then the fructose 6-phosphate is further activated by the addition of another phosphate group from hydrolysis of ATP to form fructose 1,6-biphosphate in the third stage of glycolysis. The enzyme that catalyzes reaction is phosphofructokinase. A second molecule of ATP has been consumed. The enzyme idolase split fructose 1,6-biphosphate into two sugars that are isomers of each other. These two sugars are dihydroxyl acetone phosphate and glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is the only molecule that continues in the glycolytic pathway. So the enzyme thiophosphate isomerase rapidly converts dihydroxyl acetone phosphate into glyceraldehyde 3-phosphate. In stage 6, two main events take place. First, the enzyme glyceraldehyde 3-phosphate dehydrogenase dehydrogenate glyceride 3-phosphate by transferring one of its hydrogen molecule to the oxidizing agent, nicotinamide adenine dinucleotide to form NADH. Next, glyceraldehyde 3-phosphate dehydrogenase add a phosphate from the cytosol to the oxidized glyceraldehyde 3-phosphate to form 1,3-biphosphoglycerate. In stage 7, the enzyme phosphoglycerate kinase transfer a phosphate group from 1,3-biphosphoglycerate to ADP to form ATP and 3-phosphoglycerate. This reaction yields 2 3-phosphoglycerate molecules and 2 ATP molecules. In stage 8, the enzyme phosphoglyceromutase relocates the position of the phosphate group of the 2 3-phosphoglycerate molecules from the third carbon to the second carbon to form 2 2-phosphoglycerate molecules. A mutase is an enzyme that catalyzes the transfer of a functional group from one position on a molecule to another. In stage 9, the enzyme enolase removes a molecule of water from 2-phosphoglycerate to form phosphoenolpyruvate. In stage 10, the enzyme pyruvate kinase transfer a phosphate from phosphoenolpyruvate to ADP to form pyruvate and ATP. This reaction yields 2 molecules of pyruvate and 2 ATP molecules. This is the overall reaction of glycolysis, where the final product are 2 molecules of pyruvate, 2 ATPs, and 2 NADH. That's all for glycolysis.
Following glycolysis, the mechanism of cellular respiration involves another multi-step process known as the Kreb cycle. Kreb cycle is also known as citric acid cycle or tricarboxylic acid cycle. Unlike glycolysis, Kreb cycle is a closed-loop metabolic pathway where the last part of the pathway regenerates the compound used in the first step, and it takes place over 8 different steps. The 8 different steps involve a series of redox, dehydration, hydration, and decarboxylation reactions. Now, let's look at what happened in every step together. So this is an overview showing the whole process involved in the Kreb cycle. The first step of Kreb cycle is a condensation step, combining the 2-carbon acetyl group from the acetyl coenzyme A with a 4-carbon oxaloacetate molecule to form a 6-carbon molecule of citrate. Coenzyme A is bound to sulfhydryl group and diffuses away to eventually combine with another acetyl group. This step is irreversible because it is highly exergonic. The rate of this reaction is controlled by negative feedback and the amount of ATP available. If ATP level increases, the rate of this reaction decreases. If ATP is in short supply, the rate increases. So in step 2, the formation of isocitrate is taken place. The citrate is rearranged to form an isometric form isocitrate by an enzyme ecotinase. In this reaction, a water molecule is removed from the citric acid and then put back on in another location. The overall effect of this conversion is that the hydroxyl group is moved from the third position to the fourth position on the molecule. This transformation yields the molecule isocitrate. Moving on to step 3, isocitrate is oxidized, producing a 5-carbon molecule alpha-ketoglutarate together with a molecule of carbon dioxide and 2 electrons, which reduce NAD+ to NADH. This step is also regulated by negative feedback of ADP and NADH and by a positive effect of ADP. The enzyme isocitrate dehydrogenase catalyzes the oxidation of the hydroxyl group at the fourth position of isocitrate to yield an intermediate, which then has a carbon dioxide molecule removed from it to yield alpha-ketoglutarate. In step 4, alpha-ketoglutarate is oxidized. Carbon dioxide is then removed, and coenzyme A is added to form the 4-carbon succinyl coenzyme A. During this oxidation, NAD+ is reduced to NADH+ and hydrogen ions. The enzyme that catalyzes this reaction is alpha-ketoglutarate dehydrogenase, and this enzyme is regulated by feedback inhibitions of ATP, succinyl coenzyme A, and also NADH. Now moving on to step 5, a phosphate group is substituted for coenzyme A and a high energy bond is formed. This energy is used in substrate level phosphorylation to form either guanine, triphosphate, or ATP. There are 2 forms of the enzyme called isoenzymes. For these steps, depending upon the type of animal tissue in which they are found. One form is found in tissues that use large amount of ATP, such as heart and skeletal muscle. This form produces ATP. The second form of the enzyme is found in tissues that have a high number of anabolic pathways, such as liver. This form produces GTP. GTP is energetically equivalent to ATP. However, its use is more restricted. In particular, protein synthesis primarily uses GTP. In step 6, it is a dehydration process that converts succinate into fumarate. 2 hydrogen atoms are transferred to FAD, producing FADH2. The energy contained in the electrons of these atoms is insufficient to reduce NAD+, but adequate to reduce FAD. Unlike NADH, this carrier remains attached to the enzyme and transfers the electrons to the electron transport chain directly. This process is made possible by the localizations of the enzyme, catalyzing these steps inside the inner membrane of mitochondria. Next, hydration process is taking place in step 7. The reversible hydration of fumarate to L-malate is catalyzed by fumarase. Fumarase continues the rearrangement process by adding the hydrogen and oxygen back to the substrate that had been previously removed. In step 8, which is the final step, malate is then oxidized to produce oxaloacetate, the starting compound of the citric acid cycle by the enzyme malate dehydrogenase. During this oxidation, NAD plus is reduced to NADH and a hydrogen ion. Alright, so that's it for the explanation for the add process involved in Krebs cycle. So, from the overall reaction of Krebs cycle, we can see that the net productions of this process are 2 ATP, 2 FADH, 6 NADH, and 4 carbon dioxide. And after the Krebs cycle takes place, the following reaction takes place will be the electron transport chain.
And will be continued for the electron transport chain. The electron transport chain is a cluster of proteins that transfer electrons through crystals within mitochondria to produce a gradient of protons that derive the creation of ATP. It is also called as anaerobic respiratory change, as oxygen will be the final acceptor. High-energy molecular NADH and FADH2 that produced from Krebs cycle are used to produce ATP in cytochrome. ATP is used by the cell as the energy of metabolic process for cellular function. Therefore, why electron transport chain is important. It has an important role in both photosynthesis and cellular respiration. In cellular respiration, it is the final and the most important step, as it is where the majority of ATP is created. Now let's move forward to the step in electron transport chains. It is shown as this diagram. Drop in the potential energy of electron moving downward, the electron transport chain occurs at the 3 points. The first one will be the transfer of electron from NADH to coenzyme Q. The reaction starts with NADH pass the electron while the complex I to FAN, which tightly bound to a protein. The NADH is oxidated to NAD plus and H plus. FAN pass the electron through a series of iron-sulfur complex to coenzyme Q, which accept electron one at a time, forming first the semiquinol and then ubiquinol. The energy produced by this electron transfer is used to pump proton to the cytosolic side of the inner mitochondrial membrane. As the protein falls back into the matrix through the pores in the ADP synthase complex, ADP is generated. The second one is the transfer of electron from coenzyme Q to cytochrome C. Coenzyme Q pass electron through FES central to cytochrome B and C1, which transfer the electron to cytochrome C. In complex 3, these cytochrome each contain heme as a positive group will have a different opposite protein. In the ferric state, the heme ion can accept one electron and be reduced to the ferrous state. As the cytochrome can only carry one electron at a time, 2 molecules in each cytochrome complex must be reduced for every molecule of NADH that is oxidated. The energy produced by the transfer of electron from coenzyme Q to cytochrome C is used to pump proton across the inner mitochondrial membrane. The proton fall back into the matrix through the pores in the ADP synthase complex. ADP is generated. As FADH2 is oxidated to FAD and H+. Electron from FADH2 produced by reactions such as the oxidation of succinate to fumarate enter the electron transport chains as complex 2, which contains succinate dehydrogenase. Complex 2 will transfer electron to coenzyme Q without the associated protein pumping across the inner mitochondrial membrane. Last one will be the transfer of electron from cytochrome C to oxygen. Cytochrome C transfer electron to the cytochrome A3 complex, which transfer the electron to molecular oxygen, reducing it to water. Complex IV characterize this transfer of electron. Cytochrome A and A3 each contain a heme and 2 different proteins that each contain copper. 2 electrons are required to reduce 1 atom of oxygen. Therefore, for each NADH that is oxidated, 1/2 of oxygen is converted to H2O. The energy produced by the transfer of electron from cytochrome C to oxygen is used to pump proton across the inner mitochondrial membrane. As the protein falls back into the matrix, ATP is generated. The energy generated by the transfer of electron through electron transport chain to oxygen is used in the production of ATP. Thus, the overall process is called as oxidative phosphorylation. As it involves both chemical and transport processes, hence this mechanism is called as thermometric coupling. In this process, the oxidation of 1 mole of NAD has produced 2.5 mole of ADP, whereas the oxidation of 1 mole of FAD has to produce 1.5 mole of ADP. That's all from us, and thank you for your listening. Bye bye.